Hi learners, I'm Dr. Gurunathan from Christu Jayanti College. Today we'll be discussing about very important protozoan called Giardia. And Giardia is also known as Giardia duodenalis and Giardia intestinalis. These are the common names that is given to an organism called Giardia lamblia, which is a species in the genus Giardia. And it is known to cause a disease condition called giardiasis. And this giardiasis is among 10 major enteric parasites which are affecting humans worldwide. And it is most common intestinal pathogenic protozoan in humans, and it is a recurrent parasite of other non-human species, including cattle, beavers, and dogs. This is a parasite of public health importance, and it is transmitted through several routes, including water, which can be a drinking water or recreational one, and it can be through fresh food products. When you speak about the organism or the pathogen which is responsible for the disease, it is a unicellular flagellated protozoan parasite that resides in the small intestine of the mammalian host. And it causes gastrointestinal illness, which is commonly referred as giardiasis, frequently causing mortality and morbidity throughout the world. It is a very common and may infect more than one-third of individuals in developing countries, and approximately 200 million people per year experience symptomatic giardiasis. Particularly, this is true in developing countries, whereas in developed countries, it is considered to be a re-emerging infectious agent because of its increasing role in outbreaks of diarrhea in several daycare centers, and that is making this disease a very important one worldwide. According to the protozoan morphology and host species in which they parasitize, Giardia include more than 50 species, and 5 species that are currently known are Giardia agilis, Giardia duodenalis, Giardia rda, Giardia psittaci, and Giardia muris. Only Giardia lamblia is considered to be a species which is encountered in humans, domestic and wild mammals. Coming to its taxonomic classification, it belongs to the kingdom Protista, subkingdom Protozoa, phylum Sarcomastigophora, subphylum Mastigophora, class Zoomastigophora, order Diplomonodida, family Hexametidae, and genus Giardia. Moving down to its morphological features, the parasite forms two developmental stages. One is called as trophozoite stage, the other one is called as a cyst stage. The trophozoites are usually pyriform, as you can see here on the right hand side of your screen, which has the trophozoite pyriform structures, and usually these pyriform structures are 10 to 30 micrometer long and 5 to 16 micrometer width, and they are possessing 8 flagella. Of these 8 flagella, 2 are anterior, 2 are lateral, 2 are ventral, and 2 are caudal. And they also have 2 longitudinal axonemes and 2 tangential curved median bodies. And this pyriform trophozoites, they generally have 2 nuclei with central rounded caryosome. Whereas when you speak about cyst, it is ovoid to ellipsoid in shape, and it is 11 to 14 micrometer in length and 7 to 10 micrometer in width, and it is membrane bound. And this membrane makes the organism to have a halo appearance. And this cyst is usually having 4 nuclei, either found in cluster at one end or occurs in pairs at opposite ends, and they have axonemes and median bodies. And it has remains of flagella inside the granular cytoplasm, and the cyst has a clear space between the cytoplasm and the protective cell. When coming to the host range, Infections have been recorded in many human and as well as animal populations globally, and some 40 species have been described on the basis of host occurrence, but most are morphologically indistinguishable. Three species groups have been recognized, especially on the basis of the trophozoite morphology. If you have a thin elongate trophozoites, they are assigned to Giardia agilis group. If they have short stout trophozoites, they are grouped as Giardia muris group, and these are usually attacking in the rodents, or they are usually found in rodents. And medium-sized trophozoites belonging to the Giardia duodenalis group, they are found mainly in mammals. So host specificity within each group remains contested, and epidemiological studies have frequently suggested zoonotic and waterborne transmission between mammals. Indeed, common regional names for the disease often suggest animal or water sources for human infections. Examples are beaver fever, backpackers malady, travelers diarrhea. When moving down to talk about the site of infection, flagellated trophozoites are found in the small intestines of the host, especially the duodenum. These trophozoites, which have been observed swimming within a distinct corkscrew motion in luminal content, as well as adhering to the gut mucosal surface with their ventral adhesive disc, when they detach, they leave distinct oval impressions in the microvillus layer. And this is actually the life cycle of the organism. As you can see here, trophozoites which are present in small intestine of man, they develop into cysts and trophozoites which are released in feces. The trophozoites disintegrate, whereas the cysts survive in water in the external environment. Of course, even they can survive in food. And when this water or food is ingested, this ingestion of mature cysts becomes the infective stage and they further infect the new host. And when you're speaking about the pathogenesis, the infections interfere with the normal absorptive function of the small intestine, thereby causing osmotic overload of the large intestine, resulting in watery diarrhea. Attached parasites may physically blanket the small intestine mucosa, significantly reducing the surface area for absorption. And it is also thought some parasite molecular products may exert a chemical action on mucosal cells. Infections apparently damage epithelial cells, leading to villus atrophy, which further reduces the surface area for absorption. These factors together contribute to malabsorption of fats and other nutrients, resulting in watery diarrhea and steatorrhea accompanied by dehydration and flatulence. Usually, when you speak about infection caused by Giardia duodenalis, it is a self-limiting disease and resolves spontaneously. But a few patients, they tend to persist with this infection, and that has led to chronic weight loss, retarded growth, and failure to thrive syndrome. Young individuals are most susceptible to the clinical infections, and clinical signs may remain, right? Okay, but most of them are asymptomatic in nature. Attachment to the intestinal epithelium is the primary means of infection being attacking the humans or the host cells. Epithelial dysfunction, Giardiasis is common, and electrolyte transport abnormalities is seen there. Enterocyte apoptosis and loss of epithelial barrier function is also observed, and proteinases and lectins, they also are commonly affecting the functionality of the intestinal cells. Fecal cysts can be used and it can be detected by routine coprological examinations, but test sensitivity is poor due to intermittent cyst excretion. Endoscopic techniques can also help us to identify chronic cases, particularly in detecting trophozoites in intestinal biopsy material. Sensitive and specific immunological techniques are available to detect the parasite antigens in fecal preparations, particularly coproantigen tests. Similar monoclonal antibody immunoreagents are also used to detect cysts in water samples using immunomagnetic separation techniques. With this, we conclude our session today. Thank you for your listening. Take care.